we are going to study about classical genetics. In that, we are going to study about the terminologies of Mendelian. So, Mendel noticed two different expressions of a trait. So, the traits are expressed in different way due to the fact that a gene can exist in alternate form of for the same trait, and it is called as allele. Allele means an alternate form of the same trait is called as allele. Example, tall and dwarf. If an individual has two identical alleles of a gene, it is called as homozygous. Okay, ma? If an individual is having two identical alleles of a gene, it is called as homozygous. So it is written as capital T and capital D. If an individual with two different alleles is called as heterozygous. If an individual is having two different allele is called as heterozygous. It is represented as capital T and small t. Mendel's non-true breeding plants are heterozygous or called as hybrids. Here, if the character is denoted by capital T and capital T, then it is said to be dominant character. And if it is symbolized as small t and small t, if it is symbolized as small t and small t, then the character is said to be recessive homozygous. Okay, recessive homozygous. If it is represented by capital T and capital T, then it is said to be dominant homozygous. If it is a recessive one, if it is having small t and small t, then it is said to be homozygous recessive. Mendel proposed two rules based on his observation on monohybrid cross. Today, these rules are called as law of inheritance. Mendel proposed two rules based on his observation on monohybrid cross. Today, these rules are called as law of inheritance. Inheritance means passing from one generation to the next generation. The first law is the law of dominance. And the second law is the law of segregation. These scientific laws play an important role in the history of evolution. Law of dominance. The characters are controlled by discrete units called factors, which occur in pairs. In a dissimilar pair of factors, one member of the pair is dominant and other is recessive. This law gives an explanation to the monohybrid cross. Expression of only one parental character in F1 generation and the expression of both in F2 generation. It is also explains the proportion of 3:1 obtained in the F2. Monohybrid cross. Monohybrid inheritance is the inheritance of a single character, that is the plant height. It involves the inheritance of two alleles of a single gene. In the F1 generation, all the plants were tall. In F2 generation, both tall and dwarf plant appeared. The dwarf trait disappeared in the F1 generation and reappeared in the F2 generation. The term genotype is the genetic constitution of the individual, that is T, capital T and capital T. The term phenotype refers to the observable character of an organism, that is the morphology, tall and dwarf of an organism. In a genetic cross, the genotypes and the phenotype of an offspring can be easily understood with the help of a diagram called Punnet's square, named after a British genetician Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotype of offspring in a genetic cross. Parent generation. Tall plant is crossed with the dwarf plant. In the F1 generation, all were tall plants and the F1 generation is selfed, and in the F2 generation it was appeared as both tall and dwarf plant reappeared. Here the genetic ratio is 1:2:1 and the phenotypic ratio is 3:1. Test cross. Test cross is crossing an individual of unknown genotype with a homozygous recessive. In Mendel's monohybrid cross, all the plants are tall in F1 generation. In F2, tall and dwarf plants were in the ratio of 3:1. Mendel self-pollinated dwarf F2 plants and got dwarf plant in F3 and F4 generation. So, he concluded that the genotype of dwarf was homozygous, that is small t and small t. The genotype of tall plant capital T or capital T or small t from F1 and F2 cannot be predicted. How we can tell if a plant is a homozygous or heterozygous? To determine the genotype of a tall plant, Mendel crossed the plants from F2 with the homozygous recessive dwarf plant. This he called a test cross. Test cross is used to identify whether an individual is homozygous or heterozygous for a dominant character. If an unknown character is crossed with homozygous dwarf, the offsprings of the F1 genotype is capital T and small t, and another one is small t and small t, and genotypic ratio is 1:1. And phenotypes are tall 1 and dwarf 1, that is the phenotypic ratio is 1:1. From this we can conclude that the unknown parent is a heterozygous tall plant. Next one, if a unknown parent is crossed with homozygous dwarf in the F1 generation, the genotypes are tall, that is capital T small t, and capital T and small t, and phenotype is tall and tall. So from this we can conclude that the unknown parent is a homozygous tall plant. Backcross. Backcross is a cross of F1 hybrid with any one of the parental genotypes. The backcross is of two types. They are dominant backcross and recessive backcross. It involves the cross between the F1 offspring with either of the two parents. When the F1 offsprings are crossed with the dominant parents, all the F2 develops dominant character and no recessive individuals are obtained in the progeny. If the F1 hybrid is crossed with the recessive parent, individuals of both the phenotype appear in equal proportion, and this cross is specified as test cross. The recessive backcross helps to identify the heterozygosity of the hybrid.